effects of peripheral perturbations on the central mechanisms. And then she did a postdoc in Dan Feldman's lab, where she studies EI balance in mice models of autism. So I think that's all I'll say for now, because we're extremely late. So Michelle,
okay. Thank you, Alex, for that nice introduction. And thank you, Dr. Wang, also, and Dr. Gouda and Dr. Shan for the invitation to speak today. It's truly an honor to have this opportunity. So I am the chief of the section on neural circuits at the National Institute on Alcohol Abuse and Alcoholism at the National Institutes of Health. So the section of neural circuits is a relatively new section. So we started in January of 2020. And our goal is to understand how genetic and environmental insults alter the input to computation in and the output of biological circuits to cause neurodevelopmental disorders like autism, epilepsy, and ADHD. So to fulfill our mission, SNC uses a multi-systems approach, and we address our scientific questions using primarily genetic mouse models. So as you know, nervous systems function using a balance between excitation and inhibition, and this balance is tightly regulated. I want to understand how is this balance regulated and what are the consequences of its impairment to disease. So, but in order to study these questions, we must study a bigger question, which is how is circuit homeostasis maintained in the normal brain? So mechanistically, what is homeostasis? So you can imagine that in a network. Hello?
Michelle, you're not sharing your screen?
Oh, I'm not? Oh. Oh. Okay. I don't know. Can you see it now?
I can see a little slide of it. Yeah, I can see it.
You can see it now?
Yeah.
Yeah.
Yes.
Okay, sorry, let me change. Is it fine now?
Yes.
Okay, so okay, I will start over. Okay. So here, so as I mentioned, to fulfill our mission at the SNC mission, we use a multi-systems approach, and we address these questions primarily using genetic mouse models. So as you know, nervous systems function using a balance between excitation and inhibition, and this balance is tightly regulated. I want to understand how is this balance regulated and what are the consequences of its impairment to disease. But in order to answer these questions, you must study a bigger question, which is how is circuit homeostasis maintained in the normal brain? So mechanistically, what is homeostasis? So one can imagine that in a network, you have a balance between active neurons that are firing action potentials and non-active neurons. And there is some genetic or sensory or temperature perturbation that disrupts this balance, such that the proportion of non-active neurons becomes enriched and firing rates in the network decrease. But neural networks are built to do something, so that's not a state that is allowed. So that reduction in firing rate is detected by perhaps a sensor, which sends information to the control center effector. And the net result of this is a normalization step, where the balance has been restored and stable firing rates established. So the second question that I'm interested in, which is quite unrelated to the first in some ways, is autism spectrum disorder or the occurrence of febrile seizures due to a failure of circuit homeostasis. So these two disorders may seem disparate at first, but I'll connect the dots shortly. So febrile seizures are seizures that are induced by a fever, and we know that the thermal component is sufficient. We also know that there's an age-related susceptibility, so that in humans, the peak susceptibility occurs at 18 months of age, but by 5 years old, there's very little chance of inducing a febrile seizure. For autism spectrum disorder, it is a neurobehavioral disorder. Unlike febrile seizures, it affects children. So it can be diagnosed as early as 2 years old, autism spectrum disorder. So the diagnostic symptoms of autism spectrum disorder include the display of narrow interest, repetitive behavior, social communication deficits, and abnormal response to sensory stimuli. So what connects these two disorders is the fact that they're hypothesized to be disorders of elevated activity in brain networks. So we want to understand how much of this elevated activity in brain networks could be due to a failure of circuit homeostasis. So for many of us, when we think of a parameter that is homeostatically regulated, we think of body temperature. So today, I will define a homeostatic mechanism that the brain uses to keep firing rate stable when the temperature elevates into fever range. The body temperature is maintained close to a set point by negative feedback loops. And the value of the set point, which in this example is 37 degrees, which is true for many of us, is intrinsically determined. But throughout the day, during the day, one naturally experiences minor fluctuations in body temperature from the set point. And these fluctuations are less than 0.5 degrees Celsius typically. But what's also happening in the system is that you have major fluctuations in body temperature. So here we are measuring the body temperature in 10 mice every 5 minutes over 6 hours. And the temperature is being monitored by transponders that are subcutaneously implanted in the animal. And we can see here that over the 6-hour period, the average temperature for these animals is 36.5, and we can also appreciate that the body temperature fluctuated from the set point no more than 0.5 degrees Celsius. But if we focus on within subject variability, we can see that for all 10 animals, at some point during this 6-hour period, there was a major fluctuation in body temperature of a degree or more. So in the teal-colored floating bars, we see that for some animals, this increase or major fluctuation in their body temperature during this period was even more extensive. So it rose to the level of 2 to 3 degrees. And for the animals with the speckled colored teal bars, which are animal 3 and 9, we see that the extent of this increase pushed their body temperature into fever range, which is more than 38 degrees Celsius. So major fluctuations in body temperature occur along with minor fluctuations in body temperature, but major fluctuations aren't seen to be normal, but they occur in a variety of situations. So for instance, for freely moving animals, body temperature can increase by as much as 3 degrees. From Dr. Kiatkin's work at NIDA, we know that during motivated behaviors such as social interaction, sexual arousal, eating, drinking, body temperature can increase again by as little as 1 degree to as much as 3 degrees. So not only is body temperature increasing by this extent common, but typically when we think of major fluctuations in body temperature, we think of fever. And fever, as I mentioned, is an increase in your body temperature above 38 degrees Celsius. And for most of us, when we have a fever, we actually would experience a low-grade fever, where the body temperature is elevated by 1 to 3 degrees Celsius. So the most common cause of fever is, of course, infection, but there are numerous causes of fever. And for little children, one of the most common causes of their body temperature being elevated into fever range is the fact that they're overdressed, and that impairs their ability to thermoregulate. It impairs their ability to release heat into the environment, and that results in their maintenance of heat, the retention of heat, and so their body temperature increases. What's also underappreciated is the fact that your body temperature is increasing and you're experiencing these major fluctuations in body temperature during the fever, but your brain temperature is also increasing as well. And the reason for this is because global brain temperature is congruent with body temperature. And we've known this for a while now. So this is a figure from a Holzman paper in 1981. And here they are measuring body temperature via rectal probe. And in this particular situation, they are elevating the body temperature in rats. And that body temperature is elevated by placing the animal on a hot plate. And that hot plate disrupts their ability to lose heat, so their body temperature and brain temperature are increasing. So the brain temperature measurement is being denoted by these triangles, and you can see that they closely follow each other. So in our experiments, we also measured the body temperature and the cortex temperature in awake, freely moving animals. And we see that, again, that the brain and the body temperature are congruent. So in general, you find that the brain temperature, the global brain temperature, is about 36.9, and the rectal temperature is about 36.9 as well in human beings. So what are some of the behavioral effects of increasing your body temperature and in turn your brain temperature into fever range? So in most people, it actually has no effect. You can increase your body temperature and brain temperature by as much as 3 degrees with no effect. But for some individuals, the effects are quite harmful. So in 2 to 5% of children between 6 months to 5 years of age, they experience febrile seizures. And there are also non-idiopathic cases where there are people with identified genetic mutations in certain genes like sodium channels, SCN1A, GABA-A receptor, gamma subunits, and so forth. And these people are prone to getting seizures upon their body temperature being elevated into the fever range. On the other hand, you have some individuals in the population where the rise of their body temperature into fever range is beneficial. So in 17 to 83% of autism patients, this includes infants, children, and adolescents, they experience symptom improvement. And this symptom improvement is notable because currently most pharmaceutical drugs on the market are not effective at treating the diagnosed symptoms. However, you have a case here where fever is very effective at reducing the incidence of the diagnostic symptoms. So understanding the mechanism which is promoting this is pretty intriguing. So what we're focused on at SNC is understanding why it is that you increase the body temperature in fever range, and for most people, the effects are benign. And in understanding this, we're hoping that we can in turn shed light on why similar increases in body temperature may be harmful in some and beneficial in others. So our goal now is to identify mechanisms that preserve brain function when brain temperature elevates into fever range. And for our study, our mouse experimental window is from P12 to P23. And this experimental window was chosen based on the age range where febrile seizure susceptibility has been observed in genetic mouse models. So in these mouse models, febrile seizures are induced not by an infection, but rather by exposing the animal to heated air. And that heated air increases their body temperature into fever range. So in this FGF13 mouse model, for instance, febrile seizures are observed at P15, but they're not observed at P40. So in a very common model of febrile seizures, and in general, Dravet syndrome, the SCN1A mouse model, this particular mouse model, febrile seizure onset occurs at P20. So there appears to be, at minimum, at least two distinct phases and maybe two or more mechanisms that help protect the brain from elevation of temperature into fever range. So one window could be possibly occurring before P20 and the other window after P20. But because the fever effect has been observed on individuals from 2 to 18 years, our experimental window should be able to detect mechanisms that function in the ASD condition as well. So our assessments of brain function were done in a part of the mouse brain called the mouse somatosensory, or S1 cortex. And this is the part of the mouse brain that possesses tactile input from the whisker. And the reason this particular part of the brain is appropriate is because the S1 is a very important loci in the propagation of febrile seizure activity. In terms of autism, 80 to 90% of individuals with autism have sensory deficits, which include tactile deficits. So the mouse somatosensory cortex is a great place to try to understand the mechanisms that might be relevant to the ASD condition as well. So the tactile input from the whiskers enters in layer 4 of the cortex, which has 6 layers, 5 of which are shown here. And the layer 4 input is then projected to the above layer 2-3. And this layer 4 to layer 2-3 input is critical to the intracortical processing of this tactile input. So we can remove the mouse brain and make acute slices of S1, and we could mimic the normal sensory inputs into layer 4, which will activate the circuit through electrical stimulation. So we electrically stimulate in layer 4, and we can see the resultant responses in the above layer 2-3 pyramidal neurons. So the circuit that we are studying can be really simplified to contain these 3 components. So you have the pyramidal neuron, which is receiving inhibitory inputs primarily from the PV, or parvalbumin positive inhibitory interneurons. And they're also receiving excitatory inputs from layer 4. So the excitatory inputs and the inhibitory inputs result in the influx of positive and negative charges respectively into this pyramidal neuron. And in this particular example, the net effect of that is to move the resting membrane potential from -70 millivolts to some spike threshold. So the influx of these positive charges results in brief depolarizations, which we call EPSPs, and the influx of the negative charges result in brief hyperpolarizations, which we call IPSPs of the membrane potential. And the neuron sums the EPSPs and IPSPs, and if the net depolarization is more than 25 millivolts, the cell produces a spike. So one of the first things that we did was try to understand how increases in temperature affects this process. So first, for these experiments, I want to point out that we're stimulating in layer 4, And we're recording the postsynaptic potentials and spiking at a remembering potential that is just subthreshold. And we are first recording these responses, these layer 4 evoked spikes and PSPs at 30 degrees. And this is again in acute slices containing S1 cortex. And then we increase the temperature to 36, and we make a successive measurement at 39 as well. So these are example traces of layer 4 evoked activity. And the position of spike threshold is indicated by these open circles. What you might appreciate from this example trace, and that which is summarized in this figure, is that the spike threshold, the value of the spike threshold depolarizes as you move from 30 to 36 to 39 degrees. So there's a significant increase. As you would expect, if at 39 degrees, your spike threshold is depolarizing, you'd expect that some networks would not be able to fire anymore. So the fraction of cells in a network would decrease. What was surprising is that when we looked at the number of spikes in the network that were being produced, that was constant. So this is suggesting that perhaps there is some homeostatic mechanism that's functioning in the face of a reduction in the fraction of cells spiking in the network and these depolarized spike thresholds. So we examined then the peak of the PSP, which is generating the spike. So these are example traces of the layer 4 evoked PSPs at different temperatures. And in our quantification, we didn't observe any temperature effects on the peak of the PSP. However, we did observe a positive correlation between the PSP peak and spike threshold, which suggests that in the cells where the spike thresholds were becoming depolarized, perhaps they're generating larger PSPs. We also looked at the late component of the PSP, which is enriched in the inhibitory component. And we saw that there was a reduction in the late PSP as you increase temperature. And we also saw a negative correlation between the late PSP and spike threshold, which suggests that perhaps these neurons that have an elevated spike threshold perhaps have a reduction in the inhibitory inputs. So to confirm this thinking, we then move to measuring the inputs, the excitatory inputs and the inhibitory inputs. So again, we're using these acute slices of S1. And here we are using, we're holding the cell at different voltages, the reversal potential for excitation and GABA A inhibition respectively to isolate the currents. And we also want to determine the EI ratio, which we define as the ratio of the excitatory conductance over total conductance. So in this example trace of a layer 4 evoked EPSC and IPSC, and these are convergent, we see that the excitation is reducing as you increase temperature, and inhibition is also reducing as well. But what we can appreciate from this example is the fact that inhibition may be decreasing preferentially. So quantified here, we see that from 30 to 36 to 39, we see that there is a drop. If we focus on inhibition, we see that from 30 to 36, there is a drop, and it's matched to the reductions that we see in excitation. But at 39 degrees, you have a preferential reduction in inhibition, and the net result of this is that there's an increase in the EI ratio. So one notable point is that these fever temperature-induced physiological changes are reversible. So here I'm using R series as a measure of the stability and quality of the recording, and we can see that R series is not changed. So in these experiments, we are measuring the excitatory and inhibitory currents at 30 degrees. We're gradually increasing the bath temperature to 39 degrees, and again recording the EPSCs and IPSCs, and then cooling the slice down and recording the EPSCs and IPSCs again. So in this slide, we have summarized the net effects that we see. We see a reduction in the EPSC at 39, and it rebounds to its initial values at 30 degrees upon cooling. And these are the individual cells represented here. For the iPSC, we see the same trend. So we have so far identified what may be crucial steps in maintaining circuit homeostasis in the normal brain. We see that there is an elevated spike threshold, a decrease in the percent of spiking cells in the network. We see a positive correlation between the peak of the PSP and spike threshold, increased EI ratio. And even though in the face of all of this, we end up with stable mean firing. So now it's time to try to connect these dots. So one of the first things you wanted to determine was whether either an elevated spike threshold and or decrease in the percent of spiking cells in the network was necessary and sufficient to drive an increase in the synaptic EI ratio. So to do that, we turn to the use of drugs, designer receptors exclusively activated by designer drugs. And as you know, these drug receptors are protein engineered so that they carry reduced susceptibility, binding, to their endogenous ligand, which is acetylcholine, and they preferentially bind clozapine and oxide. And upon binding to this inhibitory version of the TRED, it can produce decreased cell excitability in that cell. So what we aimed to do was at 36 degrees, where we knew the fraction of cells spiking in the network was stable, so there was no change from 30 to 36. Our goal was to activate these inhibitory drugs, and in doing that, we are hoping to see that there is a reduction in the fraction of cells spiking in the network, and in turn, that should produce an increase in EI ratio. If in fact, this reduction in the fraction of cells was necessary, sufficient, to produce an increase in EI ratio. So here we are showing the results of that experiment where we did indeed observe at 36 degrees when we added CNO, a reduction in the fraction of spiking cells. For reasons we don't quite understand yet, we also observe a depolarization of the spike threshold as well. But importantly, what we see is that upon addition of CNO, excitation doesn't change, but we do see a reduction in inhibition. And this is sufficient to drive an increase in EI ratio. So what we know now is that at least the elevated spike threshold and or the decrease in the percent of spiking cells in the network is sufficient to increase the synaptic EI ratio. What's interesting is that all these mechanisms that may act homeostatically to maintain stable mean firing rate are transient. So at P21, as the brain is maturing, it's sort of temperature proofing itself, so that at these elevated temperatures in fever range, spike threshold is no longer altered. The fraction of cells in the network is no longer changing, and in turn, EI ratio is no longer changing. But what's interesting is that even though these mechanisms are not functional in a normal animal at this age, in some autistic mouse models, at least 2 that we have observed, we do see these same mechanisms are functional. So normally in these autism mice, at 36 degrees, your EI ratio is very, very normal, and these mechanisms do not work. but in fever range, they emerge. And the example I'm going to show you today of this involves the SCN2A ASD mouse model. So SCN2A encodes the protein NAD1.2, which is a voltage-gated sodium channel. And loss of function variants of SCN2A are strongly associated with autism as well as intellectual disability. So in this example here, we see for the normal animals, as we increase temperature, we go from 30 to 36 to 39, the fraction of cells remains stable. In the autism mutant, we see that the fraction of cells as we increase temperature is decreasing. You also notice that it's elevated. For the SCN2A mutant, and wild type, respectively, you see as you increase temperature, you have matched changes in E and I, so that EI ratio remains stable. But for the autism mutants, you have a preferential reduction in inhibition, and this drives an increase in EI ratio. What's important is that spike number is relatively stable, especially in the physiological range. So to summarize, what we're seeing is at a very early period before P21 is the engagement of an EI homeostasis mechanism that in the phase of increasing the body temperature into fever range, this mechanism is activated to help keep the number of spikes in the network stable. And we think as the brain is maturing, certain intrinsic parameters of the cell become resistant to these increasing temperatures. So in a way, as the brain matures, it no longer needs these mechanisms, and it's sort of permanently protected from increases in temperature in the fever range. But in some ASD mouse models, this phenomenon where EI homeostasis is present when it really shouldn't be may underlie some of the beneficial effects that we see in autism. So right now we are focused on understanding whether or not this is true. And to do that, we are, again, switching our temperature paradigm to an in vivo approach. So we have two different temperature or thermal challenges that we give to mice. One mimics just a thermal increase in body temperature, and the other is an infection-induced increase in body temperature. And we can see from this heat map where we are measuring body temperature every 5 minutes over 6 hours. For both of these thermal challenges, we're able to boost the animal's temperature into fever range. So right now we are trying to understand whether in boosting the animal's temperature into fever range, we can indeed minimize some of the typical symptoms that we associate with autism spectrum disorder. By using some of the approaches that I previously outlined in SLICE, we're then hoping to see whether we could link the EI homeostasis changes that we see to this possible improvement in ASD behavior. So in summary, what we have observed is that for a perturbation of a cortical network, S1 cortical network, when we increase brain temperature, there are numerous changes that are occurring. We have a reduction in the layer for excitatory inputs. We also see that spike threshold in a subset of neurons becomes elevated. Now, if that's the case in this very simple example, you would expect for 4 neurons to turn into 2 neurons or spiking in the network, and that would decrease the network firing rate. However, what we are also seeing is that you have a preferential reduction in inhibition to these neurons that have undergone an increase in your spike threshold, and they have also undergone an increase in EI ratio as a result. And the net result is that instead of these neurons falling out of the network, we think they are maintained in the network so that normal firing rates are maintained. So what we are focused on understanding is the role of PV neurons. Is it more than just simply mimicking the input it is, the excitatory input? Is it doing more? Is it serving as a sensor in the network, or perhaps other roles as well? And we're also interested in understanding the molecular mechanism that may allow the PV neurons to perhaps sense the fraction of cells that are active in a network. Another one of our directions is trying to understand how much of these mechanisms we're uncovering in the mouse system may be applicable to human neurons. So we are studying this using grafted human brain tissue, which is transplanted into a mouse right now. These tissues, we find that they're vascularized, they're well vascularized, and they are multi-layer, so they extend all of the layers of the cortex, all 6 layers. And we're really trying to understand, as I mentioned, the circuit homeostasis mechanisms in human neurons using this approach. On my last slide, I would just like to acknowledge the people that are doing the work, Dr. Shen, Dr. Well, not Dr. yet, but Yadi, he's a postdoc in the lab. We have 2 new members in the lab, Jordan and Joey, and hopefully in the future you'll see some of their work. But I also like to acknowledge my support system, my former mentor, Jean, as well as members of my advisory committee, Chris Bain and Dick, Dr. Shen, and also my deputy scientific director and Dr. Kunos. So thank you for your attention, and I will take questions if there are questions.
Any questions for Michelle? Yeah, I have one. What is the source of the change in the IPSP amplitude? Is it a change in the unitary IPSP, or is it like a change in the threshold of the FS cells?
I actually don't know that yet. Whether because we're doing these experiments at P14 in layer 2-3, we haven't been able to identify the PV neurons because parvalbumin isn't actually on at that time. It's on in the deeper layers, but we don't see it in layer 2-3 neurons. So it's hard to sort of get at that question at that age. So luckily, we're starting to do these experiments at P21 in the autism mouse models, where we can use our typical tools like PV creep promoters to answer that question. What I can tell you is that we are seeing effects in other neurons, like SST neurons, but for PV neurons, I can't tell you the cause of that. But for SST neurons, we do see an increase in spike threshold as well.
And there was no change in these cells, like resting potentials? It was just a change in the threshold?
For the SST neurons, no, there wasn't a change in the resting membrane potential. But I can't speak for the PV neurons yet.
Any other questions for Michelle? Okay, here's a question. What is the evolutionary benefit for children's brains to be sensitive to temperature fluctuations?
I think it's sort of the other way around. I think evolutionary speaking, you want the child's brain not to be resistant to temperature in places, because it will be a mechanism that the brain would use, like fever, obviously for little children, is a primary mechanism that they would use to protect themselves from infection. So in these 2 to 5% of children that actually experience a seizure, they're atypical in that way. Because fever, it's supposed to normally serve, evolutionarily speaking, a protective role, and they can't even use that. They can use the fever for that role. Instead, it has adverse effects.
Okay. Another question is, how might barometric pressure influence temperature and hence neuronal threshold?
Sorry, I couldn't hear.
How might barometric pressure influence temperature and hence neuronal threshold?
I have no idea, actually. I don't know. I'm open to suggestions about that.
Okay. So the third question is, which mechanism could be behind this temperature variation? Only neurovascular coupling? Changing blood flow?
For just in general, or
yeah, what
for with fever, or
yeah, say in general.
Okay, so in general, yes. If, for instance, it's linked to behavior like increased movement, then yes, it could be metabolic. It could be also the metabolic activity being the increase in metabolic activity, and in turn blood flow can be initiated by increased neuronal activity. So you have that potential avenue. And fever, if it is due to an infection, obviously you have cytokines that are released peripherally, and that will then target the hypothalamic machinery, which will then increase your set point temperature. So it moves from 37 degrees and it's elevated, so that becomes your new normal for your body to produce heat. And another reason, obviously, is hyperthermia, where typically you would allow normal heat exchange from your body into the environment. However, if the air or the surrounding air around you is warmed, then you have a reduction in that heat exchange. And that result of that would be that your body temperature is increasing, the blood temperature is increasing. The blood temperature, as it's increased and it's going to the brain, it's no longer supplying this cool blood, which will allow the brain to eliminate its heat. So the net result of that is increased brain temperature into a fever range.
I have a question from Dick Chen. It says, intriguing new data. How do you think inhibitory neurons sense general network firing? Are there known release neuromodulators, for example?
Yeah, I think we have the typical candidates, but like BDNF and nitrous oxide. But I think what's different and why perhaps these modulators may not apply is that typically those modulators are observed for experiments with time scales for suppressed activity of 4 hours or more. Here, we're sort of looking at a much shorter time scale, talking about seconds, milliseconds. We're seeing these effects. So right now, we're not really focused on a particular diffusible factor per se, but just raw excitatory input into the network. And perhaps that might be affecting the computation within the PV neuron to affect how it reaches spike threshold and so forth. So that's kind of what we're focusing on now, looking at the inputs into the PV neuron and how the inputs are modulated and summed in the PV neuron with temperature changes.
There's a final question from Vishwa. It says, I believe Marder's work suggests some of these homeostatic mechanisms are related to channel dynamics and variability. What are the relative timescales of intrinsic homeostatic mechanisms versus more circuit-level homeostatic mechanisms?
So typically we're talking about hours, and here we're talking about milliseconds. So we're not discounting, just for the record, differences in channel gating or kinetics. Right now we're trying to narrow it down to cell type, and then the cell type obviously being the PV neuron. And then from that point, we could delve into things like what you're saying, like kinetics of channels and so forth. But we haven't reached that point yet.
Okay. Here's a question from Shai. In your in vivo model, would you be looking at the effect of sleep also?
Oh, that's wonderful. Yeah. Yeah, we actually do have plans to look at sleep as well, because I think recently there's a private company, Avison, and they did a small study, a double-blind study, where they used their device, which is supposed to mimic a low-grade fever. So the idea is to, well, I believe I have a slide on this, if I could move to it, if that's okay, or not. So the idea is that this device, once you put your hand into it, it warms the blood. And they have found that in, I think, 30 children, 30 families participated in this study. And in that very small cohort, they were able to find effects on sleep, that the sleep in the children actually improved from regular warming of their blood. And in turn, they see secondary effects, obviously, on autism symptoms, but the study wasn't powered appropriately, so there are plans to repeat it on a larger scale. So I definitely suggest that sleep may be something to look at, and we plan to follow through on looking at it.
Okay, if there are any other questions, let's thank Michelle again for giving us this excellent seminar. That's me.
Okay. Thank you.
Well, you finished on time.